Hi, everyone. I want to start by thanking Olink Proteomics for organizing this webinar and for giving me a chance to speak today. My name is Debattama Sen. I have a lab that studies CD8 T cell dysfunction in various contexts, including in the tumor. And my lab is based at MGH Cancer Center in Harvard Medical School. And today, I'm really excited to tell you about a project that's been highly collaborative with Olink, where we're trying to look at the age tumor microenvironment and understand how it limits CD8 T cell control of cancer. So, as many of us know, cancer is largely a disease of aging. We know that the risk of cancer diagnosis increases dramatically as we age, and the drivers of that are multifaceted and complex, particularly when you consider the factors in a human population. So we know that we accumulate mutations via age, and so of course there are changes in the tumor cells, including genomic alterations. We know that our immune function declines with age, and so there's an element of immune aging to consider. Other non-tumor, non-immune tissue like stroma can have aging effects. And of course, we know that there are systemic changes in aging, like changes in cytokine levels and inflammation that can affect immune outcomes. And based on the specific focus and interest in our lab, we started a project where we're particularly trying to understand how aging might limit CD8 T cell mediated control of cancer. CD8 T cells, of course, being one of the key effector cell populations that help drive anti-tumor immunity and immunotherapy responses. Because of the complexity of modeling aging and studying aging in a human population, we started with a very reductionist mouse model where we can try to isolate as many control for as many variables as possible and isolate the effect of immune aging. So in this case, we start with a workhorse syngeneic mouse model, B16 melanoma, that has been modified to express ovalbumin as a model antigen. And we simply implant those tumors into mice of various ages and look to see what happens. The first thing that we noted that's very striking is that as the mice get older and older, they are less and less able to control their tumor. And so going from blue to red, you can see a progressive increase in tumor volumes as the mice are older. And this is associated almost perfectly with drops in the CD8 T cell compartment. And so in the left plot, we're looking at total CD8s, and in the right plot, we're looking at these actual OVA-specific or antigen-specific T cell responses, but the trends are identical. And so this tells us that the aged tumor microenvironment somehow promotes tumor growth and limits CD8 T cell expansion. But of course, we know that numbers aren't the only story here when it comes to CD8 T cells, and so we want to understand CD8 function. So we look at, again, mice of these two ages, young and old, and assay first the production of effector cytokines. So here I'm showing you IL-2, interferon gamma, and TNF. And you can see that always in red, aged tumor-bearing mice have CD8 T cells that show less cytokine production. And then we can look at granzyme B as a marker of cytotoxicity and BRD urine incorporation as a marker of proliferation. And globally, therefore, we know that aging will limit the ability of CD8 T cells to produce cytokines, to be cytotoxic, and to divide. We also know that even though I have been showing this data so far in bulk, that there's actually really important heterogeneity in the tumor microenvironment within the CD8 T cell compartment. I'm showing you here an old model from a paper that we published, but this has now been shown again and again by many different groups, that there are at least two key important subsets of T cells in the tumor. These are called progenitor exhausted because they are the primary target of immune checkpoint blockade therapy, and they can give rise to the second subset, often called terminally differentiated or terminally exhausted, that is short-lived and can mediate some effector function, but ultimately has some systematic impairments. And so, of course, we wanted to know, would we see any differences in the formation of these exhausted subsets between young and aged mice? And what you might have expected going in is that if tumor control is worse in aged mice, that their T cells might be more exhausted or more biased towards terminal differentiation. In fact, as you can see in the quants showing here, that's not what we see. What we actually see is that age tumor-bearing mice have fewer of these progenitor and these T-term or terminally exhausted cells. And instead, we found an expansion of this novel subset that we had not really seen in other disease contexts that we have now started to call age-associated dysfunction, or TTADs. And then, of course, we have to ask, what are these TTAD cells? What do they represent? And so we thought one way to take an unbiased look at this is to perform single-cell RNA-seq. So the data I'm showing you here is UMAP of integrated T cells isolated from young and age tumor-bearing mice. And this is a density plot, so the brighter the color, the more there are cells in that part of the UMAP. And what you could appreciate immediately is that in young tumors, there's primarily these T progenitor and term cells and some dividing cells, whereas the tumor infiltrating CD8 T cell compartment in age tumors is substantially different. And we see, again, this strong expansion of these TTAD cells, and that's quantitated to the right. We also know that the transcriptome isn't always the best marker of cell state. And so we looked at the chromatin state or the epigenome of these cells using ATAC-seq. And so this is a PCA projection of sorted progenitor terminal, or in the case of the age tumor, also TTAD cells. And what you can see highlighted is that progenitor cells from both young and age tumor-bearing mice cluster together, as do terminal cells. But these TTAD cells really form their own group and resemble neither of these two known subsets. And so that tells us that the problem of CD8 T cell dysfunction in aging isn't about being hyper-exhausted, and that instead these cells are shuttled to this alternate cell fate that seems to be transcriptionally and epigenetically distinct from conventional T cell exhaustion. But so far, all of the work that we had done had been in the context of this very reductionist and somewhat artificial mouse model. So at this point, we wanted to know, is this subset actually relevant in human disease? Can we find these TTAD cells in human cancer? To do this, Alex and my lab compiled published single-cell RNA-seq datasets from several different tumor types, including melanoma, lung cancer, and other solid tumors. And then he did reclustering just to call different cell subsets. And we find a lot of what we would have expected. You know, we see memory-like cells, dividing cells, terminally exhausted cells, etc. But we also see this cluster in orange that we believe is the human analog of the TTAD cells we've been studying. And one of the ways that we can confirm that is just to see how those cells distribute over the age range of our patients. And so in the center density plot, we're really segregating either into 40 to 60-year-old patients or 70 plus. And you can see very clearly that in the older cohort of patients, there's a shift away from the terminally exhausted subset and towards what we are labeling as our TTAD cells in humans. And another way to confirm that this cell subset exists and is really associated with human aging is to take our mouse transcriptional signature and look for enrichment within the human T cells. And again, we can see very strongly an overlap between these mouse TTAD cells and these this human TTAD cell cluster. And so from this, we conclude that the TTAD cell formation isn't a phenomenon that's really unique to mouse models, that these cells are present sometimes in very large numbers in human tumors. And so now the question becomes, what's driving the formation of these cells in these age tumors? This ends up becoming a pretty complex question to answer because aging is such a pleiotropic process, as I highlighted at the beginning of the talk. So what we need really is a way to isolate the variable of aging either solely in the T cells to understand whether the formation of TTAD cells is cell intrinsic, or to understand the effect of cell extrinsic signals, or the TME, on this formation. And so the way we do that is by taking advantage of transfer assays. So the very top line here, what I'm showing you is the data that I showed before, which is that if we implant tumors directly into young and aged mice, we see a relative expansion of these TTAD cells and depletion of the T-term cells in aged tumor-bearing mice. But now we set up these TCR transgenics transfer so we can do, as I said, and isolate the variable of aging. So in the middle, what we have is T cells isolated either from the spleen of young or aged TCR transgenic mice, but then those cells go into a young tumor. And that would isolate the effect of aging only in a T cell intrinsic way. And then conversely, we can take young T cells and transfer them into aged or young tumor-bearing mice, which then obviously is reading out the effect of the microenvironment. And what we find is that in the cell intrinsic transfer setting, we don't see any additional formation of T-TED cells when those aged T cells go into a young tumor microenvironment. If anything, they seem to be slightly more biased towards T-term formation. But if we now look at the cell extrinsic setting, of course, the top line, which is the endogenous cells, again, we see the expansion of TTAD. But we also see that the transferred young T cells, when they go into this aged tumor, form the same cells. They're enriched for this TTAD and they're depleted in T-term formation. And that tells us that this novel subset is really not an intrinsic property of T cell aging, but is actually driven by some other signal in the tumor microenvironment. Another way to see the impact of this is by just looking at the effect on tumor growth. And what you can see is that the HTME significantly limits tumor control by these transferred young T cells. And that's partly related not just to that subset formation data I'd shown earlier, but also to their overall expansion and priming, because we recover far fewer of them from these aged tumors. So at this point, just to give a quick summary of the biology we had seen so far, there is a ever and ever more appreciated concept in cancer immunology about the cancer immunity cycle, which essentially lays out some of the key steps that have to occur to mount an effective anti-tumor response. And what I've shown you so far is that at least on the last steps of the cancer immunity cycle, the recognition and killing of tumor cells by CD8 T cells is impaired in aging. But we also see some defects in priming and expansion. And so that leads us to ask, are some of the earlier steps, such as steps 2 and 3, where we have APCs like dendritic cells driving the activation of T cells, are those affected? And so we wanted to ask how aging affects DC differentiation and function in tumors. Again, we return to our simple model where we have progressed mice of different ages and we implant B16 tumors. I'd already shown you that this is associated with ever increasing tumor volumes, but it turns out it's also associated with lower and lower numbers of these CDCs, or conventional type 1 dendritic cells. And if anything, a relative increase of the CDC2s. And so given how important these CDC1s are in activating CD8 T cells, we then looked at some of the molecules that are required to mediate DC T cell crosstalk. So here I'm showing you in those same tumors expression of CD40 and CD86, which you can see is lower more and more in progressive aging. And these are key co-stimulatory molecules, and suggests that actually there is this broader defect in CDC1 T cell crosstalk with age. So just to again contextualize in the cancer immunity cycle, what we're now showing is that some of these early steps of antigen presentation and priming and activation are also impaired in aging. And so the question becomes, what are the elements of the tumor microenvironment that's driving all of these defects? Some of these defects in the DC function activation, and obviously the defects in the CD8 T cell killing. So we went back to the different components that we're already aware of in the tumor microenvironment. And one of the things that really caught our attention is the idea that there might be these global or systemic changes in the milieu of these mice. And so we worked with Olink Proteomics to try to understand first cytokine changes in the aged tumor microenvironment. To do this, we take our same cohorts of tumor-bearing mice of various ages, and we collected tumor interstitial fluid, and with the help of Olink, enumerate the levels of many different cytokines and chemokines. Just almost as a positive control, we already knew, just based on our T cell analysis, that there were reductions in effector cytokines like IL-2, interferon gamma, and TNF. And you can see the correlate of that in terms of reduction of those cytokines in the tumor interstitial fluid. So that's great. But what we also now were able to identify for the first time is that there are other defects, particularly in chemokines for recruiting immune populations in the HTME. So CXCL9, shown in the bottom left, is really key for recruiting different T cells and other effector cells, whereas CCL4 and CCL2 are really important for recruiting myeloid cells. And we can see clear progressive decreases with age in those cytokine levels. And to convince you that this effect that I'm calling related to the HTME is truly specific to the tumor, we also profiled serum with the help of Olink from the same mice. And you can see that CXCL9 doesn't necessarily have global defects in the aged mice. In fact, if anything, maybe there's very slight increases that are non-significant. Whereas in the serum, we do see increases of other expected changes like type 1 interferon, which has been well established in the aging field as a driver of a phenomenon called inflammaging. So this gives us confidence that the changes we're seeing both in the TME and in the serum should match expected biology. And again, just to confirm also that the CCL2, CCL4 levels aren't lowered systemically. And so that tells us that at least part of the issue in the HTME is that there's a global reduction, not just in the cytokines that help T cells kill tumors, but also in the chemokines that help recruit different immune cells, particularly myeloid cells, into the tumor in the first place. And so we wondered if we took a therapy that's myeloid targeted, like CD40 agonism, could we actually rescue some of these defects and reinvigorate this whole cancer immunity cycle by targeting these upstream DCs? So it's the same setup, but now we either give control treatment or CD40 agonist antibody to these mice. And as one would expect, if we just look at young mice plus or minus CD40 agonism, there's an increase in tumor control. There's an expansion of CDC1s, particularly in tumor draining lymph node, as well as an expansion in CD8s. The aged mice, critically, also respond to CD40 agonism. And in fact, it normalizes the tumor volumes to close to what it is with the young mice. And this is, again, associated, and very importantly, associated with increases in the CDC1 frequencies in the tumor draining lymph node, as well as increases in the CD8 T cell compartment. And so that tells us that something that can really target very upstream in terms of the defects we're characterizing by reinvigorating the myeloid response can actually rescue CDC1s and enable priming and expansion of endogenous CD8 T cells. And our data, again, generated in collaboration with Olink, suggests that the chemokine defects we had characterized before are really critical to this process, because now I'm showing you those same 6 cytokines and chemokines, IL-2, interferon gamma, and TNF on the top, and the recruiting chemokines CXCL9, CCL2, and CCL4 on the bottom. And you can see that CD40 agonism almost always rescues the levels in both young and aged mice, and in some cases, such as with CCL2, and CXCL9 really brings the levels up to what would be physiological in a young tumor. And so that's very exciting in terms of giving us new approaches to try to rescue aging defects in thinking ahead towards something more translational. And finally, because we had started this from the concept of impaired DC T cell crosstalk, we wanted to make sure that the improvements weren't just at the level of recruiting more DCs, but also their activation state. And you can see that after CD40 agonism, that purple bar really substantially goes up compared to the red bar. And so these aged CDCs have better expression of class 1 and can present antigen better, and have higher expression of FLT3, which just helps recruit and differentiate more DCs. And so our data now clearly shows that the HTME disrupts multiple stages of the cancer immunity cycle to ultimately limit CD8 T cell control. We know that without any intervention, that there are defects in CDC1 differentiation and activation of CD8 T cells. Based on the work done in collaboration with Olink, we now know that there are defects in the recruitment and trafficking of different immune populations to the tumor microenvironment, and that ultimately then this prevents recognition and killing of tumor cells. So really quickly, I already gave a summary of our data showing that changes in the HTME reduces CDC1 numbers, which then leads to reductions in CD8 T cell priming, effector function, and alters their differentiation to form this novel TTAD dysfunctional cell state. And we know that myeloid-targeted therapies like agonistic CD40 antibody can help rescue some of these defects. While CD40 agonists have not had much success in translation to the clinic, we believe that our data provides proof of concept for a new approach where we both take into account the effect of aging on anti-tumor immunity and also can start to take a look at other strategies that might reinvigorate different aspects of the cancer immunity cycle to try to rescue these defects. And so in our future directions, we're really interested in dissecting DC T cell crosstalk in the HTME. While I didn't focus on them in this talk, we're also very interested in T cell intrinsic defects that are associated with aging. And then we are, of course, interested in the impact of aging on different immune-targeted therapies like checkpoint blockade, but also new and emerging cell therapies like CAR T cells and TIL therapy in solid tumors. And so with that, I will end by acknowledging my lab, who obviously did all of this work and just have been really like a powerhouse in driving this project, and also our collaborators and our funders. And thank you for your attention.